This is ComD 400, Lecture 7. Lecture 7 deals with the auditory neuron and with the auditory pathway. That is to say, how do we get information out of the inner ear and up to your central nervous system and your brain for processing and for our ability to interact with the environment, with the acoustic information that's in the environment. In this way, this lecture series, this lecture slide set, corresponding to learning objectives 197 to 224, presents some detailed anatomy, and in lecture learning objectives 221 to 222, in that range, we have to deal with the idea of, well, why is this system so complex? Why is it so complicated? And the fundamental answers to that is because we need to have sort of two different ways of reacting to or responding to processing information, acoustic information from the environment. One way is we need a very fast way to respond like animals to things defensively, to escape and to protect ourselves from things that might be dangerous. We also need to have a pathway that gets information up to our brains so that we can have many of the higher level experiences that we have with acoustic information. So spoken languages being sort of one of those, music being another. So those two have very different sort of pathways in a sense that one is shorter and one is longer and more complicated. But we have to have the capacity to do both. And so that's what we'll talk about. One of the other features, by the way, of in my mind anyway, of Lecture 7 is to present you with some anatomy that will be useful to you in other courses. And that's just some basic sort of neurology type of anatomy. So that's what we're going to continue to work through. So let's get started with the slide set here. There'll probably be a couple of slide sets, maybe two, maybe three. I'm not sure at this point. We'll see how we do. So first, as we move along, and by the way, these slides correspond with the PDF slide set that's available to you on Moodle. So I'm just moving along through those. So first question, of course, first concept we have to think about is, what is a neuron? A neuron simply is a fancy name for a nerve cell. And when we think about this idea of a neuron, the neuron is the functional unit of the nervous system, and it has basically three parts. There's a cell body that has a cell nucleus, and there's extensions of it, which in general terms we can call processes. In other general anatomy and general biology classes, you might have seen these referred to in a basic neuron as an axon and a dendrite. So basically, when we think of the three components of any neuron, we're thinking those are being sort of a dendrite, a cell body, and an axon. And so that's a component of any individual neuron. Now, when we have this other name of a term called a nerve simply is a bundle or a bunch of many individual neurons. So in this particular slide, this particular illustration, you're seeing that that there's let me find one neuron. So basically, here's sort of one neuron right through here. So there's one neuron, and here's another one, and here's another one, and that's wrapped up in a bundle. And then there's another one. There's another bundle of many neurons here, and another one there. And all of this here is what we might call a nerve. And so they're all kind of wrapped together, but you can have many neurons inside of one nerve. That's the basic idea. In this particular view from an author by the name of Haynes, here's that same view that sort of that we looked at on this previous slide, where you've got, again, many individual neurons. So there's a neuron there's another one, there's another one, there's another one. So all these different neurons are inside these subunits, if you will, of the nerve. They're all wrapped inside a bigger wrapping. So they're just sort of, we go from sort of, if you want to start at the smallest level, smaller, and then bigger units, and then a big unit like that. And again, you can see the picture here and what we're looking at. When you actually look at a photograph of that, you can kind of see the same thing here, broken down in terms of the outer layer of this stuff, the sort of the lining, and then you finally get down to the individual cross sections of the neurons themselves. When we think about the parts of a neuron, a generic neuron that might be found sort of anywhere in the body, there's some basic parts. That's the way it goes. The cell body is one of those, and the cell body is also known as the soma, the cell body. In addition to that, we've got parts that get information sort of to and through the cell body. So one of those processes that the information travels along, the first one is a dendrite. So the dendrite sort of picks up the information and then takes it to the cell body. All right, and so the information that's traveling through and across the neuron, for lack of a better word for the moment, we're just going to call it a nerve impulse. It's a little burst of electricity, so to speak, and that's picked up initially at the dendritic portion, takes it toward the cell body, and then moves that out away from the cell body by something called an axon. So in that context, then we've got sort of three things. We've got a cell body, a dendrite, and axon. If we just kind of look at it, if you will, as it might attach to the auditory hair cell. So here's an auditory hair cell, and then there's the dendritic portion here that's taken the information to the cell body, and then that takes information away. The information is always traveling this way. So here's your dendrite process, here's your cell body, and then this would be your axon. And then this would connect up to something else over here. It might be another neuron. It might be ultimately the final termination of this of the neuron. But that's the basic flow through, is always unidirectional from whatever the source is, in this case auditory hair cell, picked up by a dendrite. This area here we call the synapse, and then that information flows to the cell body, away from the cell body along the axon, and then to some other location. Here's just a picture out of the cycle book, and the cycle book has other chapters besides the auditory ones that deal with sort of the basic neurology that underlie that supports communication, speech, language, hearing. And so this is what we might refer to, and he refers to, as a generic neuron, in the sense that in a generic neuron, and this is a point of distinction, as we'll see in a moment, a generic neuron has a short dendrites. Here's the cell body. Remember, your cell body's other name is a soma. And it may have multiple impinging dendrites that all go to a cell body. But relatively speaking, the dendrites are very short relative. Remember, where the process is, the information is flowing this way. The spark is flowing that way. The nerve impulse is going that way. So you've got some information flowing in through the dendrites and then flowing out along the axon. So the axon is all of this. And so relatively speaking, if here's the dendrite portion, here's the cell body, and here's the axon aspect, the axon is much longer in a generic neuron than the dendrites, and that's typically the case. When we think of what we're calling this sort of generic neuron in the part of the nervous system that we're going to call the peripheral nervous system, and for the sake of a basic definition, the peripheral nervous system is everything that's outside of the central nervous system. And basically, so we think about the central nervous system is basically your brain and your spinal cord. So brain and spinal cord would be the central nervous system, and everything else is the peripheral nervous system. So when we talk about neurons in the peripheral nervous system, their cell bodies have a very specific name. So one individual cell body in a peripheral of a neuron in the peripheral nervous system is called a ganglion, and the plural of this is ganglia. So this is kind of a helpful locator word for the most part, that when we see this word ganglion, we have a sense or we have knowledge that we're in the peripheral nervous system. That's the basic clue there. On the other hand, oh, sorry, I wrote over that. On the other hand, this same basic structure, same basic fundamental thing when we're in the central nervous system is called a nucleus, and the plural is nuclei. Now, this is a little troublesome because when we consider this same word, nucleus is also the part of an individual nerve cell, or of any cell really has a nucleus. That's where the DNA is. But this is sort of a different use of the word nucleus, nuclei. So again, in the cell bodies of neurons in the CNS, are called nuclei. The purpose of a neuron is to provide a pathway to get a nerve impulse from something to someplace else. And again, it's processing through the central nervous system. The central nervous system has as its fundamental distinction is that information travels via electrical signals. And as I've mentioned to you in class, electrical signals really are carried through chemicals. So if you don't have chemicals, you're not going to have electrical signals. So they're intrinsically related, but when we think about what the nervous system does is it provides information, transmits information via electrical signals. The electrical signal that we're basically talking about, the primary electrical signal we're talking about, is something referred to as an action potential. And that's what any individual neuron does, is it creates and transmits a nerve impulse called an action potential. And as I said just a moment ago, these potentials, and potential, another name for a potential, as I mentioned in class, is voltage. So a voltage is just an electrical spark, an electrical impulse, and so that electrical impulse is mediated is possible because there's chemical changes that are occurring between cells. So if we go kind of back to the fundamental issue here of auditory system, we're talking about the idea that we're trying to trying to get information to your brain. So trying to take environmental information and get it to your central nervous system so that you can have a capacity to interact with that. So as we said in lecture 6, the primary sensory transducer in the auditory system are the hair cells in the inner ear, in the organa corti. And again, as we said, these are primarily the load of sensory transduction comes to the inner hair cells as compared to the outer hair cells. So their job is to take the information that they get, the analog of the environmental signal. And we say analog because the environmental stimulus of sound has been converted ultimately into fluid waves and into mechanical activities which disrupt the inner hair cells and create a chemical reaction in the inner hair cell. But the net result, the net output of the hair cell is a neuron firing at the base of the hair cell. That neuron firing now is picked up across many neurons and taken into the central nervous system. When we think about what are the two primary characteristics of the acoustic signal in the environment, so we say what happened acoustically in the environment, the two things that we're fundamentally interested in is how much sound was there, and we can call that intensity. Perceptually, we might call that loudness. And frequency. What are the speeds of the vibrations there? And again, perceptually, we know that as pitch. So we need to get, as part of this, we need to have these kinds of things get to our brains. I don't have, I think, any specific learning objectives anymore on these next two slides, 10 and 11, but just for completeness, indulge me here. We've said that frequency basically is encoded by that tonotopic organization, the place of maximum displacement along the basilar membrane. And so this notion of tonotopic organization begins in the inner ear along the basilar membrane, but is found in all nuclei. So it's preserved all the way through the auditory system, even up to the auditory cortex. In other words, that there is a specific relationship, a spatial relationship of frequency arrayed along a tissue. So primarily that's how tonotopic, that's how we get frequency information, is by where in the specific receptor area what is actually being stimulated, what's responding. The other sort of feature that we're looking at is this notion of intensity. And intensity basically is coded by the number of auditory neurons that are involved and by how fast they're firing. So the basic idea is that any one neuron can only fire, can only create an electrical stimulus only so quickly, because if you think about it for just a moment, what's happening with the neuron is that it's a chemical reaction. The chemicals involved in creating the spark and creating that auditory impulse, the electrical impulse, is requires chemicals. And so with one impulse, then the chemicals have to be restocked and resupplied, and that takes a little bit of time. Not a lot, but it takes some time. And so there is a limit on how fast any given neuron can fire. Generally said, the louder the stimulus in the environment, the faster a neuron is going to fire. That's what the system is reading, is the speed of the neuron firing, and we equate that perceptually with the loudness at the nervous system level. But again, any individual neuron has a limit on how it fires, and so what then is involved is multiple nerve fibers get involved in order to accommodate higher firing rates, meaning in order to encode and get information that we would understand as being higher amplitudes or louder signals. Again, I don't believe there's any learning objectives on these. All right, so when we think about, though, again, what's the purpose of the primary auditory neuron? It's to get the information, the electrical impulses that began at the base of the cochlea, and get them initially to the brainstem, and then ultimately to higher processors in the central nervous system. The auditory nerve is the last structure, or is that bridge structure of the peripheral auditory system. So the auditory nerve begins in the inner ear and ends in the brainstem, and the brainstem will then be part of the central nervous system. So this auditory nerve that we're going to talk about, again, is this bridge structure that starts in the PNS, in the peripheral nervous system, and ends in the central nervous system. And as we say here, and as we talked about already, the auditory nerve, that first auditory neuron, begins at the base of the hair cells, travels out of the organocordy, cordi, out of the scala media, into the medullus, and then out of the medullus to where it finally, that initial primary auditory neuron terminates at the first structure in the central auditory nervous system, in the central nervous system, in the brainstem, at a group of nuclei called the cochlear nuclei. Now, this is kind of a weird name because the cochlear nuclei aren't really in the cochlea; they're actually in the brainstem. But they're the first group of nuclei that are in the that exist after the primary auditory neuron. Okay, so on that, I want to conclude this particular screencast, and we're going to jump to screencast 2, where we'll focus really on the auditory nerve, primary auditory nerve. See you in a little bit.